Now in this video we are going to see the classification of plasma cell dyscrasias. What are plasma cell dyscrasias? These are a group of disorders characterized by the abnormal proliferation of plasma cells. What are plasma cells? They are specialized P lymphocytes. The function of plasma cells is they produce immunoglobulins. Now what happens in plasma cell dyscrasias? There is overproduction and secretion of abnormal monoclonal antibodies or their fragments. These are called M proteins or paraproteins. These disorders can be benign, they can also be malignant. Now let's see what are the different type of plasma cell dyscrasias are seen. Now we see what are the different type of plasma cell dyscrasias. They may be benign, as we already mentioned. One of them is monoclonal gammopathy of unknown significance. What happens is this: the serum M proteins are less than 30 gram per liter, with less than 10% of the bone marrow population is plasma cells, and there is no evidence of bone destruction or tissue damage. And then we have asymptomatic multiple myeloma. In this asymptomatic multiple myeloma, or sometimes also called smoldering multiple myeloma, there is high protein, that is more than 30 grams, and the plasma cells are more than 10% of the bone marrow population, but there is no evidence of bone destruction or tissue damage. And then we've got plasmacytomas. Plasmacytomas are discrete solid plasma cell tumors. They may be found in bone or they may be extramedullary in soft tissues. The immunophenotyping and genotyping abnormalities may be similar to multiple myeloma. And then we've got primary amyloidosis. There are intact light chains or heavy chains fragments, immunoglobulin, forming beta amyloid sheets which are deposited in different organ systems, causing damage to those organ systems. And then we've got malignant disorders. One of them is classical multiple myeloma. The M protein concentration is more than 30 grams and the plasma cell population in the bone marrow is 10% or more. There is evidence of bone and tissue damage. So this is malignant myeloma. Then we've got plasma cell leukemia, which is a very uncommon disorder. And you see more than 2000 plasma cells are present in the circulation, or 20% of the circulating pool of white cells is plasma cells, or more than 20%. So these are malignant disorders. The benign entities like asymptomatic multiple myeloma may degenerate into malignant forms. So one has to keep a watch and follow up. And then staging of multiple myeloma. There is an international staging system, that is ISS. We've got 3 stages. Stage 1 is serum beta-2 microglobulin is less than 3.5 milligram per liter, while the serum albumin is equal to or greater than 3.5 grams per deciliter. Stage 2 is neither stage 1 nor stage 3. It's different from these. The stage 3 is serum beta-2 microglobulin is more than or equal to 5.5 milligrams per liter. And then we got revised international staging system. This includes some chromosomal features. The stage 1 is as international staging system stage 1, but there is standard risk chromosomal abnormalities by FISH and serum LDH at or less than upper limit of normal. So serum LDH may be normal or less than normal. Stage 2 is not revised international staging system 1 or 3. And then we see what is the stage 3. International staging system stage 3 and either high risk chromosomal abnormalities by FISH, for example deletion 17p, translocation 414 and or translocation 1416, or serum LDH level is greater than the upper limit of normal. So this is, we see the staging system of multiple myeloma.